Today's topic is development of T cells and B cells. The major cells in the adaptive immunity are the lymphocytes. The lymphocytes include the T cells, B cells, and the natural killer cells. Of this, the B cell and T cell possesses the characteristics of diversity, specificity, memory, and self-non-self recognition, the hallmarks of an adaptive immune response. In the bone marrow, hematopoietic stem cells are present, from which all the cells of the immune system are derived. Hematopoietic stem cells grow and mature on a meshwork of stromal cells that support the growth and differentiation of hematopoietic cells. Initially, the hematopoietic stem cells differentiate into lymphoid progenitor cells and myeloid progenitor cells. The lymphoid progenitor cells further differentiates into T cells progenitors or pro-T cells and B cell progenitors or pro-B cells. The maturation of pro-T cells to functional T cells takes place in the thymus, and the maturation of the pro-B cells to the functional B cells continue in the bone marrow. B cell development in the bone marrow. The generation of matured B cells first occur in the embryo and continue throughout life. Before birth, the yolk sac, fetal liver, and the fetal bone marrow are the major sites of B cell maturation. After birth, generation of mature B cells occurs in the bone marrow. B cell development begins as lymphoid stem cells differentiate into the earlier distinctive B lineage cells, the progenitor B cells or pro-B cells, which expresses a transmembrane thyroxine phosphatase called CD45R. Pro-B cells proliferate within the bone marrow, filling the extravascular spaces between large sinusoids in the shaft of the bone. Proliferation and differentiation of pro-B cells into precursor B cells or pre-B cells requires the microenvironment provided by the bone marrow stromal cells. Stromal cells include fat cells, endothelial cells, fibroblasts, and macrophages. Stromal cells influence the differentiation of hematopoietic stem cells by providing a hematopoietic inducing microenvironment consisting of a cellular matrix and factors that promote growth and differentiation. Many of these hematopoietic growth factors are soluble agents that arrive at their target cells by diffusion. Others are membrane-bound molecules on the surface of stromal cells that require cell-to-cell contact between the responding cells and the stromal cells. The stromal cells play two important roles in B cell development. They interact directly with pro-B and pre-B cells, and they secrete various cytokines, notably interleukin-7, that supports the developmental process. Major steps in B cell development. The maturation of B cells involve the following major steps. Step 1. At the earliest developmental stage, pro-B cells require direct contact with stromal cells in the bone marrow. This interaction is mediated by several cell adhesion molecules, including VLA4 on the pro-B cells and its ligand VCAM1 on the stromal cells. Step 2. After initial contact is made, a receptor on the pro-B cells called c-KIT interacts with the stromal cell surface molecule known as stem cell factor. This interaction activates c-KIT, which is a thyroxine kinase, and the pro-B cell begins to divide and differentiate into pre-B cell and begins expressing a receptor for interleukin-7. Step 3. The interleukin-7 secreted by the stromal cells drive the maturation process, and the pre-B cells differentiate into immature B cells displaying membrane-bound immunoglobulins. Eventually, the maturation process induces downregulation of the cell adhesion molecules on the pre-B cells so that the proliferating cells can detach from the stromal cells. At this stage, pre-B cells no longer request direct contact with stromal cells, but continue to require interleukin-7 for growth and maturation. Immunoglobulin gene rearrangement in B cell maturation. B cell maturation depends on rearrangement of the immunoglobulin DNA in the lymphoid stem cells. First to occur in the pro-B cell stage is a heavy chain DH to JH gene rearrangement. This is followed by VH to DH-JH rearrangement. If the first heavy chain rearrangement is not productive, then VH-DH-JH rearrangement continue on the other chromosome. Upon completion of heavy chain rearrangement, the cell is classified as a pre-B cell. Continued development of pre-B cells into an immature B cells require a productive light chain gene rearrangement. Because of allelic exclusion, only one light chain isotype is expressed on the membrane of a B cell. Completion of a productive light chain rearrangement commits the now immature B cells to a particular antigenic specificity determined by the cell's heavy chain VDJ sequence and light chain VJ sequence. Immature B cells express membrane IgM on the cell surface. The bone marrow phase of B cell development culminates in the production of an IgM-bearing immature B cells. At this stage of development, the B cell is not fully functional. Full maturation is signaled by the co-expression of IgD and IgM on the membrane. This progression involves a change in RNA processing of the heavy chain primary transcript to permit production of two mRNAs, one encoding the membrane form of the mu chain and the other encoding the membrane form of the delta chain. Thus, IgD is a characteristic cell surface marker of matured naive B cells. B cell selection in bone marrow. It is estimated that in the mouse, the bone marrow produces about 50 million B cells per day, but only 5 million or about 10% are actually recruited into the recirculating B cell pool. This means that 90% of the B cells produced each day die without ever leaving the bone marrow. This loss is attributable to the negative selection and subsequent elimination or clonal deletion of immature B cells that express autoantibodies against self-antigens in the bone marrow. T cell development in the thymus. Progenitor T cells or pro-T cells from the bone marrow begin to migrate to the thymus for maturation at about 11 days of gestation in mice and in the 8 or 9 weeks of gestation in humans. T cell maturation involves rearrangement of the germline TCR genes and the expression of various membrane markers. In the thymus, developing T cells known as thymocytes proliferate and differentiate along developmental pathways that generates functionally distinct subpopulation of mature T cells. All developing T cells also undergo a process of thymic selection before they go out into the circulation. Steps in T cell maturation. Step 1. After arriving at the thymus, pro-T cells enter the outer cortex and slowly proliferate. Differentiating T cells progress through a series of stages that are marked by characteristic changes in their cell surface phenotype. For example, thymocytes early in development lack detectable CD4 and CD8. Because these cells are CD4 negative, CD8 negative, they are referred to as double negative or DN cells. The initial thymocyte population displays c-KIT, the receptor for stem cell growth factor, and CD44, an adhesion molecule involved in homing. CD25, the beta chain of the interleukin-2 receptor, also appears on early stage DN cells. Step 2. The DN cells stop expressing c-KIT, markedly reduce CD44 expression, turns on expression of the recombinase genes REC1 and REC2, and begins to rearrange their TCR genes. Initially, the DN cells begin to rearrange the TCR beta chain genes and start expressing the beta chain. Newly synthesized beta chains combine with a 33 kDa glycoprotein known as the pre-T-alpha chain and associates with the CD3 group of molecules to form a noble complex called the pre-T cell receptor or pre-TCR. Step 3. Formation of the pre-TCR complex gives the following signal. Indicates that a cell has made a productive TCR beta chain rearrangement and signals its further proliferation and maturation. Suppresses further rearrangement of TCR beta chain genes resulting in allelic exclusion. Renders the cell permissive for rearrangement of the TCR alpha chain. Induces developmental progression to the CD4 positive and 8 positive double positive state. Step 4. After advancing to the double positive or DP stage where both CD4 and CD8 co-receptors are expressed, the thymocytes begin to proliferate. However, during this proliferative phase, TCR alpha chain gene rearrangement does not occur. Both the REC1 and the REC2 genes are transcriptionally active, but the REC2 protein is rapidly degraded in proliferating cells, so rearrangement of the alpha chain genes cannot take place. The rearrangement of alpha chain gene does not begin until the double positive thymocytes stop proliferating and REC2 protein level increases. The proliferative phase prior to the rearrangement of the alpha chain increases the diversity of the T cell repertoire by generating a clone of cells with a single TCR beta chain rearrangement. Each of the cells within this clone can then rearrange a different alpha chain gene, thereby generating a much more diverse population. Thymic selection of T cells. The most distinctive property of mature T cell is that they recognize only foreign antigens combined with self-MHC molecules. To achieve this, the developing thymocyte undergoes two selection processes. Positive selection for thymocytes bearing receptors capable of binding self-MHC molecules, which results in MHC restriction. Cells that fail positive selection are eliminated within the thymus by apoptosis. Negative selection that eliminates thymocytes bearing high affinity receptors for self-MHC molecules alone or self-antigen presented by self-MHC, which results in self-tolerance. Thymic stromal cells including epithelial cells, macrophages, and dendritic cells play essential role in positive and negative selection. These cells express class 1 MHC molecule and can display high levels of class 2 MHC also. The interaction of immature thymocytes that express the TCR-CD3 complex with populations of thymic stromal cells result in positive and negative selection. Positive selection takes place in the cortical region of the thymus and involves the interaction of immature thymocytes with cortical epithelial cells. Cells whose receptors are not able to bind MSC molecules would not interact with the thymic epithelial cells and they die of apoptosis within 3 to 4 days. The population of MSC restricted thymocytes that survive positive selection undergo negative selection by interaction with thymic stromal cells. During negative selection, dendritic cells and macrophages bearing class 1 and class 2 MSC molecules interact with thymocytes bearing high affinity receptors for self-antigen plus self-MSC molecules or for self-MSC molecules alone. Cells that experience negative selection are observed to undergo death by apoptosis. Tolerance to self-antigens encountered in the thymus is thereby achieved by eliminating T cells that are reactive to these antigens. T cell development is an expensive process for the host, as estimated 98% of all thymocytes do not mature. They die by apoptosis within the thymus either because they fail to make a productive TCR gene rearrangement or because they fail to survive thymic selection. Double positive thymocytes that express the TCR-CD3 complex and survive thymic selection develop into immature single positive CD4 positive thymocytes or single positive CD8 positive thymocytes. These single positive cells undergo additional negative selection and migrate from the cortex to the medulla where they pass from the thymus into the circulatory system. As a conclusion, I would like to say that development of B cells and T cells is crucial for the immune system as they play an important role in the adaptive immune response. B cells and T cells also bear the major antigen recognizing molecule, immunoglobulin and T cell receptors. They have all the characteristics of the adaptive immunity and this is achieved by a stringent developmental process. The developmental process ensures to produce diverse B cells and T cells to recognize the millions of antigens that are encountered every day. Again, the selection process during B cell and T cell development ensures that the B cells and the T cells are unresponsive to self-antigens which may otherwise cause deleterious consequences to the host. Though the developmental process is expensive with only 5% to 10% survival, the final product are the robust and diverse B cells and T cells which compensate the expenses.